Hello friends, today I welcome you all for the discussion of this beautiful experiment of physics in optics, that is Edser A diffraction pattern. First of all, you can see the experimental setup in the figure, that is one telescope, the Edser plate, the wire gauge, the sodium lamp and all the apparatus which we will be discussing right now. But before going further, we first of all look forward at the aim, which is to find the wavelength lambda of light using Edser diffraction pattern. And the apparatus used for this experiment would be Edser A plate, the light source, which is sodium, the telescope, the traveling microscope, and the plate stand. The preferable environment, friends, here would be a dark room. But in case there is no dark room at your lab, then you can even perform this experiment at a room where the light is very less. Okay. So first of all, our first apparatus, that is the light source, which is very important part of an experiment, particularly in optics. The light source, as you can see here, is the sodium lamp. Sodium lamp is a monochromatic light, which means monochromatic, that is single color, or in advanced terms we can say single wavelength. Friends, you very well know that each and every color has its own wavelength, right? So here what we can say is this particular sodium light has a single wavelength. As far as the sodium light is concerned, actually it is having a sodium doublet, that is lambda 1 lambda 2, but these two colors are very near to each other, that is lambda 1 and lambda 2 are very close to each other. So for general case, we cannot differentiate both of these doublets easily, and therefore it is also called a monochromatic light. Fine. So that is our first apparatus, that is the light source. Next, as our name of the experiment suggests, Edser A diffraction pattern, we need a plate where you can see this alphabet A. Friends, it is very interesting that why the alphabet A only? Why cannot I use any other alphabet? Your initial or my initial. Can we use it or not? The answer is yes. You can use any alphabet that you want, any shape that you want, and light will do its job, right? Whenever light actually falls on an obstacle, it deviates its path, right? And this bending is called the diffraction. Of course, the process of diffraction is successful only at that time when the incoming light wavelength, that is lambda, matches with the order of the slit that is open. That is the pure definition of diffraction. Otherwise, even in a big room having two windows, you can say that the double slit experiment can happen. Actually, some way or the other way, the diffraction is happening, but it is not able to be seen. That is, it is not observed easily. Why? The reason is the window has a width of say 3 meters or 4 meters, and the incoming light, say for example sunlight, is of some angstroms, that is 10 raised to minus 8 centimeters or 10 raised to minus 10 meters. So the order has a wide difference. Okay, so for these reasons, successful diffraction is very much dependent on the slit width. And as far as this experiment is concerned, you can even use any other alphabet and you can definitely get the diffraction pattern. Okay, so first of all, let us switch over to the figures that we have here. The first figure is from the lab manual diagram, where you can see the diagram of A and you have to measure the distance between the 2 points, that is the crossbar measurement of A. The second figure is the same figure, that is an analogous diagram representing the same. And the third figure is the actual Edser A plate. Friends, you can even make this Edser A plate by your own. You can take an X-ray and with a blade you can just, with a very fine hand, you can like crave that A here. Or as you are seeing here, I have taken a white cardboard and with a blade it has been carved here, right? So Edser A plate is very simple. Now, the next thing is the distance between its two crossbars has to be measured with the help of the traveling microscope. It requires a very fine measurement. Why? Because this fine measurement will let you go for the accurate results. Okay, so this measurement I will show in the next slide, but it will be named as P. The distance between these two crossbars will be P. Next important thing, because of which the actual diffraction is more effective, is the wire gauge. See friends, wire gauge is shown in these two figures. There are two different kinds of wire gauge here. One is having big squares, other is having very minute squares. Please ignore the middle part, it is actually ruptured, but you can see that this portion has very minute squares. Now again, this particular wire gauge, we have to calculate the distance d between the vertical lines or two horizontal lines. Since they are squares, it is the same. If you measure the distance between two vertical lines or two horizontal lines, the distance will be the same. Since it is the square, all sides are equal. Again, my friends, this distance is measured by the help of traveling microscope. I hope you must be familiar with the traveling microscope setup. You can see a lab setup design here, where first of all I have put on the wire gauge, and the second plate you can see the Edser A plate for measuring the distance between the two crossbars. So what I will do, with this screw, I will make the traveling microscope move on this particular optical bench, and this is the reading table from which I will get the readings. For this screw, it is the screw for the adjustment of the height of the this microscope, and this particular setup will let you get the distance between the two horizontal lines or two vertical lines. Fine. So till now we had three setups. First of all, the light, which was monochromatic. Second, the Edser A plate, and third was the wire gauge, and of course the traveling microscope as well. Now, how to perform, how to start the experiment? What we will do, first of all, we will turn on the sodium lamp. Please note it, guys, whenever you will turn on the sodium lamp, it will take time to get 100% intensity, right? So just turn it on and take readings after 2 or 3 or 5 minutes, say for example. As in the figure, you can see that it is completely enlightened and 100% intensity is observed in the lamp. Fine. So in this way, you need a plate stand where you will fix your plate just to meet the height adjustments. As such, it's not necessary, but to meet the height adjustments, you will fix the plate in this way, and this is a closure view. Students and friends, you can see here that I have fixed this plate in an inverted position. Now, why have I kept this so? You can see the reason in the next slide. Whenever you see this from the telescope, which is kept at a relatively far distance from the Edser A plate and the source setup, that inverted A now seems to be straight. The reason is very obvious, friends, because many of you have observed many things from the telescopes or even binoculars or many other things. So that inverted object has come to be straight now, and the photo which you are seeing right now, I have tried to click it from my phone, so it not might be that much clear, but still you can observe that if you will observe this from the telescope, you will see such a pattern, right? You can very well recognize and observe the Edser A here. Right now, you are not observing any diffraction, mind it. Now what will happen? Okay, before adjusting the wire gauge, what you have to do is mount the wire gauge on a stand and place it in between the plate A and the telescope. Say that's your source, this is your Edser A plate. You have a particular distance here, then you place your wire gauge and then your telescope, and this is the place from where you will view it. One thing is very important in this experiment to get good results is all the instruments should be collinear to each other and at the same height. Viewing height should be same. If anything is not aligned, you will never get pattern. And mind it students, if you will do it accurately, you will get the results at the first itself. But if you will not align the instruments properly, you might not get the results anytime. So this alignment is very necessary. First thing collinear, and second thing the height, right? We are moving on stepwise. First of all, we took all the apparatus, we arranged it accordingly in a collinear and a height, and then we will actually start observing. But what is to be observed? Friends, you already saw in the previous slide that from the telescope you have such kind of view, right? Just a pattern. Now, how would the view change when you have inserted the wire gauge here? So let's get ahead. After keeping the wire gauge, what you need to do is try to set the telescope, try to focus the telescope and change the distance d. What is d? D is the distance between the wire gauge and the Edser A plate. You can very well see in the figure that this is your wire gauge, this is your Edser A plate, which is the inverted A, and the distance between these two objects is the distance d. What you need to do is you need to change this distance d simultaneously viewing in the telescope, right? It is not that one is viewing from the telescope and other is adjusting. Of course it can be done, and if the distance is far more, then we need to take off help of the second friend, but these two processes should go simultaneously. The only reason is, it is not like once you come and adjust the wire gauge and then you come at the telescope and again view it, right? So basically you need to adjust your wire gauge in the forward direction or backward direction, and it would be like something like this. Okay, so what will happen in this whole distance d? You will have 2 points. I'm just talking right now about 2 points where you will observe the first order diffraction pattern and the second order diffraction pattern, right? Now what will happen? What is this diffraction pattern? What is this order? And why only two positions? See friends, I have named these two positions as D1 and D2. You will observe the first order diffraction pattern at distance D1, which you have to calculate, right? You need to do some mechanical work and then calculate that distance where you will observe the first order. But how does this first order look like? First of all, I should know what I want to see. Otherwise, I will not recognize it. So first of all, we shall try to understand what is this first order diffraction pattern and the second order diffraction pattern. So the next slide is the first order diffraction pattern where n is equal to 1 is the first diagram. Will show you the lab manual diagram where you can see that before keeping the wire gauge, you only had the A in the dark line, which you can see here. You have named it as the 0th order. But since now you are putting the wire gauge before it, the diffraction will be observed. And you can easily see that a symmetric pattern will be on the right hand side and the symmetric pattern will be on the left hand side. Totally symmetric pattern, and you will see multiple images of A. But friends, these images has some symmetry, and that symmetry will help you to recognize the order. Say for example, in the second diagram, I have tried to make an analogous pattern or a diagram with PowerPoint tools, that how exactly you will see through the telescope, right? Please concentrate here. Since the background is black, your light is yellow, I have chosen the color yellow here. Of course, you need to understand that you will not observe any dotted lines from the telescope, right? You will only observe these dark lines, same dark lines. But why I have kept these lines as dotted? This is just to differentiate the first order or the second order from the original 0th order, right? Now friends, you can see a beautiful pattern here. Since it is for alphabet A, what happens is it is displaced this side, it is displaced that side. Actually, it is not displaced, it is the order of the diffraction, but it seems to be displaced, right? So what happens, it is displaced in such a way that in this triangle of A, what you have is, friends, this is just a technique or a way to remember. Again, I am repeating, for the first order, in this triangle, we have a diamond shape in the middle and two small triangles around it, right? So if you see this pattern through telescope, that means you have achieved your first order diffraction pattern. And this is the actual view from the telescope. Of course, I had some camera limitations, and still I tried to take a photograph from the telescope lens. So you can see that it is a very hazy view, but still you can observe that this is a diamond shape that I am observing, and these are the two triangles, and we have multiple images of A. These multiple images of A are due to the wire gauge, right? So this is your first order diffraction pattern. Now let us study the second order. The second order is not much different from the first order, but it is complex, right? It is not easily obtained because since the first order had a very simple figure in the triangle, we just had a diamond inside and the two triangles around it. But in the second order, what happens is we have four more small diamonds in that big diamond and two small triangles and two trapezium like shapes in the roundabout triangles. So what happens is this particular pattern is really difficult to observe. For this you need a really sharp wire gauge, really sharp your Edser A plate. The darkroom should be completely dark, the source should be actually bright, your telescope should have very good resolving power, and of course your instrument handling capacity and the way you perform experiment, all the things need to fall in right place. So if the things are green, that is if the things are working at its own pace in a good way, then you will easily see the second order diffraction pattern. Again, here I have tried to capture it from the camera, the second order. You can just compare it with the previous figure. I had here some clear vision of the first order, but somehow could not get the second order by the camera, mobile phone camera. But still, I was able to see it in my lab, and you can see it in this way. The same pattern will be observed. Students, this is a very important point. When you write in your journals or manuals, please compare these two diagrams and then draw it in your journals, right? You have to actually make difference between the first order where the figure is very simpler and the second order where it is a bit complex. So the thing is, the distance d at which you are getting the first order is your d1. That is, d1 is the distance between the wire gauge and the Edser A plate, d1, which lets you get the first order. And at the same time, d2 is the distance again between the wire gauge and the Edser A plate, which lets you get the second order diffraction pattern. So you can see the formula in both the slides. For first order, we had lambda is equal to p. Remember students, p was the crossbar measurement of A. Small d was the distance of the vertical lines and the horizontal lines from the wire gauge. N1 is the order 1, d1 is the distance which you calculated. The second order also has the same formula, only the things that will change here is n2, which will be 2 here, and d2, which will be your distance here. So with these things, we can make our observation table. You can just put on first order here, second order here, you can mention the distance here, and you can calculate lambda. Now students, what happens is I have not mentioned the result here, but actually the result is very clear. If you are taking the sodium light, yellow light, the result should come around 5893 angstroms, right? In both the cases, you will get the same results. This point is very important. Look friends, the order may change, the diffraction order may change, but the wavelength of light is not changing. So what is changing? Whenever you are changing the order, its distance is changing, right? So it is like in a relationship. If one parameter is increasing, other is decreasing. If other is increasing, then the first one is decreasing. So somewhere balancing its original value. So that value is lambda, and which is supposed to come constant around 5893 angstrom, or if you want to convert it in terms of centimeters or meters or nanometers, you can definitely do it. So one more thing I want to say, that is a hint, that whenever you want to calculate lambda, you can also do it in a different way. That is, many times students try to find out some other way to simplify the experiments, and that is not at all wrong. What you can do is, you already know the answer here, lambda 5893. You can place this in the denominator, and you can make d as the formula maker. In this way you can verify that your answer is correct or not. And whenever you will get d1 and d2, you will observe that the value of d, that is the distance between wire gauge and the Edser A plate, for the first order would be more, almost double, as compared to that of n is equal to 2. So that's all how the practical goes. We will again revise it. What we did is, we first of all selected the source, that is the um, source sodium lamp. Second was the Edser A plate. Third was the crossbar, that is the wire gauge. After that, we arranged it, mounted it here. Then we saw it from the telescope. Now we brought into picture the wire gauge. We adjusted it. We changed the distance. Then what happened? We got two orders, first order and the second order. We mentioned in the observation table, and then finally calculated lambda, that is the wavelength of sodium light. Right? So I hope this experiment was theoretically and practically clear to you all. And thanks for watching. And I would really acknowledge my college HVPGR Kadi, because the instruments and the lab setup that I have used in this presentation belongs to the Department of Physics, HVPGR Kadi, and HVHP Institute of Postgraduate Studies and Research Kadi. It is a constituent institute of Kadi Sarva Vishwavidyalaya Gandhinagar. I welcome you all for studies and any other campus visit. Thank you.